Hi, 12th grade biology students. Today we're going to be talking about what's been on everybody's mind lately, infectious diseases. So, the topic is 6.3, defense against infectious diseases, and here we go. So, there's been many diseases throughout time. We're going to go through a few examples first. So, malaria, which is a disease caused by a parasite transmitted by the bite of an infected mosquito. Ebola, which is a virus. So it's different than malaria in that it's not a parasite, it's a virus. And in 2014, there started to be a very large epidemic of Ebola spreading throughout different parts of Africa. Obviously, the current one, coronavirus, COVID-19, that we've been focusing on the most lately. And I wanted to go through this page really fast just to give you some ideas of what it is. So obviously this virus you've heard a lot about, so some of this might be repetitive. But a lot of things that we've talked about or that you might hear about in the media is it's being compared to a flu virus because technically it is a version of the H1N1 flu virus. It's similar in its shape and its structure, but obviously different, because it's a different strain of the virus. So this virus will cause a mild to severe respiratory illness. How the virus spreads isn't yet fully known though, and it's likely spread through what they believe is contact of bodily fluids. So like sneezes, and then when it lands on something and you touch it and then you touch your face. Its incubation period is 1 to 14 days. So the symptoms that they notice are common for COVID-19 would be a cough and a fever. But here you can see the comparison of the symptoms to a cold and a flu. So the main two that seem to be dominant is fever and cough. And then obviously if you've been near someone who's had it or potentially coming from a place where someone has had it. You can see here the difference in the severity of it. So this is stating how many people will be infected by an average individual with the disease. So the flu is usually 1.3 people, where COVID-19 is 2 to 2.5 people. The incubation period is much longer, so quarantine is important, staying inside, since it's 1 to 14 days before you start showing the symptoms. And then you can see the percentage of hospitalization rate from this, as well as cases of fatality rates is a little bit higher for the COVID-19. So when we talk about disease-causing organisms, it's not just viruses. So there's bacteria, for example, E. coli, protozoa, or like the malaria parasite, viruses, which would be the flu, HIV, COVID-19, and also fungal types of diseases like athlete's foot. Bacteria are usually prokaryotes that cause diseases. They don't have a real nucleus. Remember, prokaryotes do not have membrane-bound organelles. They divide by binary fission, so they make lots of copies of themselves. They can cause food poisoning like salmonella. You can get ear and eye infections. This is an image of someone with pink eye, or conjunctivitis, which is a form of a bacterial infection. And cholera, or diarrhea, which is most commonly known, can be a bacterial infection. Viruses are acellular, so we consider them non-living because they need to have a host in order to carry out their functions of life, including reproduction. So we don't consider viruses a living thing. They can have DNA or RNA, depending on the type of virus. Usually it's RNA. They mutate and evolve and recombine very quickly. Some of the things they've caused are the flu, or HIV, or AIDS, smallpox, measles, the common cold, herpes, Ebola, and COVID-19. This is a picture here of the Spanish flu, or the 1918 flu epidemic, which killed between 17 to 100 million people. This was a lot of people during that time. That was a quarter of the this was a lot of people during that time. They believe about a quarter of the world population, which was about 500 million people, contracted this flu, and there was about 2 billion people during that time. So 500 million people contracted it, and about 17 to 100 million died. This was also this was the first of us seeing the H1N1 influenza virus. It later resurfaced in 2009 during that flu pandemic. It's called the swine flu pandemic. They believe up to 1.4 billion people contracted the swine flu, but the deaths was far less. It was around 150,000 to 500,000 people who died. This is another type of disease, a fungal infection. So usually these are eukaryotic types of cells, and they reproduce with spores. So here what you're seeing is athlete's foot. So ringworm would be an example of this, or mold. They can also cause allergic reactions and respiratory problems. So the great thing about bacterial diseases is that we have antibiotics that can kill them. So what happens with antibiotics is they are taking advantage of the differences in eukaryotic cells. They disrupt the way that the metabolism of a bacterial prokaryote or eukaryote, so that it can't reproduce and it can't do many of its metabolic pathways. And so that kills those bacterial cells and you recover. So antibiotics are for bacterial infections only. Florey and Chain's experiments. This has to do with penicillin. Fleming was the person who discovered penicillin. It was an accident, actually. And so you can see here, this is a petri dish with this type of bacteria growing on it. And a mold started to grow on the petri dish, and that mold was releasing penicillin. And what they noticed was this zone of inhibition right here, you can see, where the bacteria couldn't come near the mold. So the mold, or this fungus, was protecting itself, and they realized it was releasing penicillin. So that was Fleming, but Florey and Chain did experiments to actually see if penicillin could help cure people after they've been infected. So here in this picture, they did a lot of tests with mice. You can see 8 different mice. In the control group, they did not use any treatments of penicillin, and in the experiment with mice, they did penicillin injections. The result was, in the control group, all 4 mice died. In the experimental group, all 4 mice survived. So they were the first to actually attempt to use penicillin to cure bacterial infections. Some bacteria, though, unfortunately have antibiotics. So there are bacteria that can survive antibiotics, and then they, while all the other bacteria died, these bacteria survived. They then reproduce, and that passes on their genes to the next generation. And then we have many bacteria who now are resistant to that antibiotic. So this is the problem with bacteria and how fast they can change and mutate. So just to kind of compare the differences, viruses versus bacteria, you're going to see here the virus comparison versus bacteria. So like we said before, bacteria can have antibiotics and it can kill them. Viruses, you cannot use antibiotics. That will not kill a virus. Because viruses do not have a metabolism, antibiotics do not work on them. So if you notice here first the size, bacteria are usually a bit larger. Bacteria have a cell wall where viruses don't. They have a protein coat. Bacteria do have DNA in loops, the plasmid loops, where usually viruses don't have DNA, they have RNA. Bacteria do not have a nucleus because they're usually a prokaryote, so they have a nucleoid, the DNA loop, and they don't have a nucleus, no organelles. And then viruses also do not have a nucleus. The RNA is floating around freely. And then on the cell surface, the viruses have spikes that can allow them to puncture through a cell wall, and they can invade, where a bacteria usually has flagella, which is a long tail, or pili, for movement and recognition of things around them. So why don't antibiotics work against viruses? So this is what I had just mentioned in the last slide. Viruses have different metabolic pathways to bacteria, so the antibiotic drugs have nothing to target. So here you can see the shape of a virus. You can see the capsid head. This is called the sheath. And then these would be the pins that they can use in order to invade into another cell. So here's a picture of multiple viruses trying to invade this cell, and they're injecting their RNA into that cell. And they want that cell to start, they use that cell's DNA replication methods to make copies of its RNA. So our first protector from diseases, infectious diseases, whether bacterial, viruses, parasites, or funguses, is our skin. So the skin is continuous and it's hard to find an opening. So there's many layers also to our skin. There's not just one thin layer, there's multiple tough layers. The skin is dry, usually. pH of our skin is not favorable to pathogens. It's not an environment that they can be successful in. We also have lysosomes within our skin cells, which these enzymes can break down pathogens before they get in. And then natural organisms. So it's competitive exclusion by non-harmful microbes. So we do have some things living on our skin naturally, is what this means. Here are the openings that potentially could lead to a pathogen entering our bodies. So the reason why they would be, within those openings though, there are also protective barriers. So there's a sticky mucus which can trap invaders. The pH is not very favorable, so like in our nose, our mouth, our trachea. And then lysosomes, so enzymes will break down pathogens in this area as well. So kind of switching, another way that things could get into our skin though is through cuts, right? So what our body tries to do is protect us by blood clotting. So firstly, not releasing the blood, but also the sooner it clots, the less likely pathogens can get in. So like it says here, cuts can provide a pathway for pathogens to enter the body, but clotting is going to seal the cut to prevent entry. Platelets release clotting factors, which are then going to close that hole. This is called a cascade reaction, which what's happening is fibrinogen, which is soluble in the blood, is going to be converted to fibrin, which is insoluble, so it becomes like a solid barrier, by an enzyme called thrombin, which is going to trigger this reaction to happen. The clot is formed in 3 to 6 minutes, and I want you to watch this video in order to understand what's happening.
In a first step towards clot formation, platelets are recruited to the site of vessel injury by now exposed molecules of the vessel wall, such as collagen and von Willebrand factor. This factor mediates the linking of platelets to collagen via a specific receptor in the platelet membrane. The resulting change of shape of the platelet from its resting state into the dendritic form indicates activation. The activated platelet in turn releases prothrombotic molecules, such as adenosine diphosphate, ADP. By binding to its receptors, ADP induces aggregation. And recruits further platelets to the site. Thromboxane is another important mediator of platelet activation and aggregation. Under its influence, the platelets cross-link with each other. These interlocking mechanisms cause platelet activation to snowball. The clot grows rapidly. Activated platelets also trigger the coagulation cascade and thus the formation of thrombin. Thrombin, in turn, stimulates platelet activation even further, a continuous feedback loop. Additionally, thrombin induces the formation of fibrin for the mesh stabilizing the clot. The self-reinforcing process of platelet activation, crucial in the formation of blood clots, is an obvious therapeutic target in conditions caused by inappropriately triggered blood coagulation.
Okay, so next, what happens when pathogens get in? So at some point they will get in, and our body has an immune system response that is very specific to attacking and remembering these pathogens. So phagocytic leukocytes, which is a non-specific immunity. We have two types, non-specific and specific. I'm going to focus on first the non-specific immunity. This is when a new invader gets into our body, a new pathogen has entered. So a phagocytosis is an eating cell. Sometimes you'll hear them referred to as a macrophage, like a big cell that just likes to eat. And then you'll also hear of a leukocyte, which is a white blood cell. So these are the first reactors to a new invader or a new pathogen entering the body. So the first step would be chemotaxis, which is the movement in response to chemicals. So what happens is it attracts the phagocytes to the area of invasion as a response to a protein that's produced by the pathogen. Usually it's called an antigen. Phospholipids released by the damaged cells. So the damaged cells release these phospholipids, and it triggers the cell to realize something's wrong. The phagocyte is going to attach to the pathogen cell surface, and then it's going to engulf it. So it's going to attach itself to it, and then it starts pulling it in and breaking it apart. A phagosome is a vesicle that contains a pathogen's form. So a phagosome is going to be filled with lysosomes, which deposit their enzymes inside of it, and they will help break down the pathogen, and then they usually excrete it in a process called exocytosis. So that's the first step for non-specific immunity, so that the body can help attack something it doesn't recognize. After that occurs, the body goes into a process of trying to make antibody production. This process is to help remember if this invader or this pathogen ever comes back and able to attack it faster. So an antibody is a protein we produce in response to a specific type of pathogen. And the antigen is a cell marker or protein. So the antibodies, it's kind of like a puzzle piece. The antibody is going to have the correct shape to fit to this antigen that came from the pathogen. So for example, if you look at our blood types, I'm blood type A, and I have anti-B antibodies. So that means my body's looking for, if it ever sees type blood B enter my body, it can attack it because it has these memory cells or these antibodies to attack that shape. So if you notice, it matches the B shape. So someone who has type B blood has these antigens on their blood cells. So mine would be more circular. Notice that someone who has type B blood has the antibodies for type A blood. So they are the same shape as these. So if they ever enter this body, the antibodies can attach to it and attack it and start breaking them down before they circulate in the body. Someone with type AB blood does not have any antibodies because their body accepts both types, so they don't need them. Someone with neither antigen A nor B, this would be someone with type O blood, notice there are no antigens. This is why they're the universal giver, but they have antibodies for both A and B. So they will attack any A or B blood that enters their body. So universal giver, universal taker. So now the specific immunity response. So this is what happens when our body remembers a pathogen that has once been there. So first, a specific antigen is identified. So here is that shape, that protein coming from that pathogen. And here is the helper T cell that remembers it. Okay, so we call it a specific B lymphocyte. It's a type of white blood cell. It is identified, and it produces the antibody that will bind to the antigen. So here's the antibody binding to the antigen. Then clonal selection occurs. This is when the B lymphocyte starts to make many copies or clones of itself. So here you can see there's many clones of it. And the idea is that now that there's many of them, they can go and attack any antigens that have entered the body. So we call these memory cells. So that's specific immunity because it's memorized this antigen and it's able to attack it much faster than it would have if it was the first time it entered the body. So this picture is showing the non-specific reaction, which is here, and then this would be showing you the specific immunity reaction here. Okay, so our last part we need to talk about is HIV and AIDS. So HIV stands for human immunodeficiency virus, and AIDS stands for acquired immune deficiency syndrome. Usually, or someone starts with HIV and it moves towards AIDS by the end of its cycle. So how does one get HIV? It's through bodily fluids that are exchanged either by sex, reusing unsterile syringes. So this would any of the fluids that the syringe has touched in your blood, or blood transfusions. So it's bodily fluids. It's not through kissing, for example. It wouldn't be saliva. It's fluids that are from blood or sexual activity. So what HIV does, what this virus does, is it recognizes a certain protein in the cell membrane of a helper T cell. Now, helper T cells are very important with your specific immune response. They're the ones who start the clonal selection, making copies of the helper T cells that need, or killer T cells that need to go out and kill the virus that has entered your body. So helper T cells help communicate which cell needs to undergo cloning processes and begin antibody production. So without these helper T cells, which HIV attacks, it weakens the immune system. So if you look at this graph here, you can see what they call the viral load. So this is what they would measure if someone has HIV. So they're measuring how much of the virus is within the host, and they're also measuring how many CD4 lymphocyte cells they have, which is the helper T cells. So you can see at first, when someone's infected, you have a lot, this is the blue line, of helper T cells. And then you have less copies of HIV, of the virus. But over time, this is probably the peak here, where you're feeling the most symptoms. And then it starts to decrease. This is the clinical latency period. So this is where the immune system, because it's a slow process. AIDS doesn't usually come till later, years after HIV develops. So this is the body fighting each other. So this is your helper T cells trying to attack it, and you can see they increase because they killed some of the virus. But because your immune system is not making as many helper T cells, eventually there's a critical point where they meet, and there's going to be more HIV than there are helper T cells. And at this point here, we would say when you have less than 200 helper T cells, you have now contracted AIDS. And usually death is soon to follow. So looking at HIV, because this was something that in the 80s became really prominent, and it was looked at as a very confusing thing. So I want to look at the big picture here. So who's more likely to contract HIV? So HIV is transmitted through blood or sexual contact, body fluids, placental fluids, childbirth, and breastfeeding. So if a woman who is pregnant has HIV, she's most likely going to pass it on to the child.
Here's what I found.
Distribution, transmission, and is uneven around the world. If you notice, it's not everywhere, but it's in certain populations. Why? So transmission risk is increased depending on the society's traditions, beliefs, and behaviors. So where in one culture, it might be okay for a man to have many wives, and if he has HIV, he'll pass it to many of the wives. Or in a society where rape is more common and there's no actions taken to stop a rapist, then potentially he can spread it to many women in his community or men. Also, we noticed within a drug behavior society, that's why in the 80s it started to boom a lot. There was a lot of transmission of it through needle sharing. So rare minority of individuals do not have cell receptors though and do not develop AIDS. So they have found there are a few people who are immune to HIV, that the virus cannot infect them. Condoms and latex barriers only protect against transmission through sexual contact. So safe sex practices will protect against HIV, but not obviously sharing of needles and things like that. So some social implications of HIV are, once someone has contracted HIV, there are some things that start to occur socially. So treatment's really expensive, so that's going to affect their life. If they wanted to get treatment, that can help reduce their symptoms. It's very, very expensive treatment. It's not proven to cure, but it can increase the years of your life. Discrimination against victims. People often have to tell if they have HIV for example, in schools, because the school needs to be aware if that student gets cut, how the nurse needs to make sure they're protecting themselves. So in some places they don't have to tell though, and because they want to protect that person's privacy, to make sure there is no discrimination against that person. Moral obligation of wealthy countries to help poorer countries. This is a topic that, if you think about, it's spreading through some areas in Africa, and they are less developed countries. And what does that mean for wealthier, more developed countries? Do they need to have a role in helping stop this or break this cycle in these poorer countries? Economic consequences. Because they're going to be becoming sick, and they become sick more often, they can't go to work as often, and there's a loss of wage for these earners. There's an increase in the number of orphans because a lot of the parents, whether, let's say, the husband has it and he passes it to his wife, and now there is an orphan left behind. So there's been an increase in the number of orphans in Africa because of HIV. Traditions, beliefs, and behaviors affect the response as well. For example, in Swaziland, having sex with a virgin can cure your HIV, is what they've been told, or is what the community belief or tradition is. So then that's going to just lead to another person getting HIV, because scientifically that would not cure HIV. This topic is actually something very near and dear to Miss Dr. Jaramillo and my heart. This is a school in Uganda. It's called the Nyaka's AIDS Orphans Project. All these students you see right here are orphans because their parents have both died of AIDS. And this was a school that our friend Jackson, so you can see this book here, A School for My Village, that he built because he wanted to educate the students there and to break this cycle of AIDS that was going through his village. If you read the book, you'll learn about his story and why he was so motivated for it. I put the link in the actual lecture if you would like to look at the project and understand it a little more. But All of these students, the goal is that they're educated and that they will, instead of staying in the village, they can go off to university and they can hopefully bring back to the village a different economic level, and that they can bring their education and they can help the people that are there and help break this cycle of passing HIV and AIDS through their community. There's another program that's been going on and is connected to this as well. It's called the EDGA Foundation, that is to help with young girls who are survivors of being raped, which is also a reason why HIV has been passed through the community. So take a look if you're interested, but this is a project that's very, very important to us, and it's one that's helping break this cycle of HIV and AIDS in other countries, in Uganda particularly. Okay, that's it. So if you have any questions, let me know. If you could please upload your notes that you took to ManageBack so I can keep track of and give you points for the MinPlus portal. Thank you.